Dan and I are part of a grant at Western to extend the capabilities of a personalized medicine lab. A lab is capable of genotyping patients before starting on drugs where genetic variation is known to cause adverse events or limit drug effectiveness. A natural question that arises in this setting is how much drug can I give the patient so that the patient remains safe, but the drug is still effective? The lab doesn't have unfettered access to patients. They can only sample them once or twice. So we think this is a good opportunity to use a Bayesian method with good priors so that dose selection models that we might develop don't have to rely solely on the data collected from the lab alone. When looking at the pharmacokinetic literature, we found that papers which claim to use a Bayesian approach often used maximum a posteriori, or MAP. That's certainly one way to compute the posterior, but over the last 10 years or so, open source tools which use Hamiltonian Monte Carlo or HMC have become more popular. We wanted to compare MAP and HMC directly for personalized medicine applications. In short, our paper contributes three things. One, a new Bayesian model for Apixaban pharmacokinetics written in STAN, an open source language. Two, a simulation study demonstrating that inferences via MAP and HMC can lead to very different dosing strategies. And three, an induction dosing model for Apixaban based on desired trough or max concentration levels after a first dose. We use data collected from our clinical pharmacology colleagues to fit a model. We fit a one-compartment pharmacokinetic model to Apixaban concentration data from 36 patients measured 8 times across 12 hours. Once we fit the model, we use the posterior to generate pseudo-patients for a simulation study. We then fit an updated model to the simulated data using both MAP and HMC, and compared predictions of latent concentration as well as uncertainty estimates for the latent concentration. We find that when it comes to predictive accuracy, MAP and HMC perform very similarly according to popular loss functions. But estimates of uncertainty between each model can vary quite a lot. Figure 5 in our paper, shown here, showcases 19 pseudo-patients in which MAP's credible interval for the latent concentration is 1.5 times as wide or wider than HMC's at its widest point. This difference in uncertainty manifests as a large discrepancy in dose size selection and consequently in measures of calibration in a Bayesian decision-making framework. We conclude that though MAP can be fast and is similar to maximum likelihood, we recommend that practitioners use HMC or at the very least fit both models in order to detect any differences in uncertainty estimates. Our code and data are available on GitHub. You're free to clone our repo and reproduce our analysis. Thank you.